Welcome, AP Biology, to our Chapter 15 discussion on gene expression and gene regulation. We're going to be breaking up our talk into two major sections for prokaryotic cells and eukaryotic cells because they're going to differ quite a bit. So let's start off with prokaryotes, so our bacteria and archaea. And how are they going to do it? Well, because there aren't as many membrane-bound organelles and not as much happening on the inside of the cell, we're going to have some simpler rules here. It's important to note that transcription and translation occur nearly simultaneously. So as you see in the picture here, as mRNA is being built off of transcribed off of the DNA, it's immediately entering into these ribosomes to make that protein. And so gene regulation is only really going to occur by turning transcription on or off. And then they're going to use promoters, which are little sections in the DNA that signal the binding of RNA polymerase to begin transcription. So that's going to be where we start from. So a big component of this is going to be using feedback. So negative feedback regulation of as things are made, those products then blocking the production of that same protein. And we're going to be focusing on something called an operon. Now, an operon is a group of metabolically related genes that share a single promoter or a single region that's going to tell all these different genes to get transcribed at the same time. Metabolically related means these genes, or the proteins that these genes make, probably work together for some common function or common purpose. These are really common in prokaryotes, and we're going to look at two different types: inducible and repressible. And as we're going to see, what happens is we make some protein, and then these products turn off gene expression. So the presence or absence of a molecule that controls transcription of an operon control the access of RNA polymerase. And so there's going to be something called a repressor protein. What this repressor protein does is it's going to turn off the genes. It's important to know that operons are always going to be repressed. There's nothing that activates or turns it on. So operons get turned off. They are not turned on. And that's going to be really common throughout the living world for how these systems work, is that we frequently are turning things off, not turning things on. And if things are getting activated, what's actually happening is they're being blocked, and then by turning them on, we're removing the blockers. So inducible operons, their default state is off. And so what that means is that our repressor protein here is binding to our operator in the promoter region, and it's permanently bound. So when it's bound, our RNA polymerase can't bind to the DNA. It can't start transcribing. So in our default state, our genes are off. And a good example of this is the lac operon, which is involved in lactase. So this lac operon, when lactose is absent, there's no lactose present in the system, our repressor is active and it's binding to the operator, just like we saw earlier. Now, when lactose is present, we need those enzymes to utilize lactose. So what happens is that there's going to be lactose is going to bind to the repressor. And when it does that, it causes a conformational change that's going to deactivate that repressor. Now that repressor can no longer bind to the promoter region. It cannot block RNA polymerase, and RNA polymerase is going to be able to transcribe all those genes that are going to make the enzymes we need in order to break down lactose. Now, eventually, all that lactose will get broken down, and there'll be no lactose to bind to the repressor protein. And at that time, when all the lactose has been used up, these repressor proteins will rebind to our operon and shut it off, so we won't make the enzymes we need for lactose anymore. So whenever a operon is blocked by a repressor, that means DNA transcription cannot occur. You can think of it as completely shutting off access to the DNA. And so our RNA polymerase can't actually read it. When there's a lot of lactose, however, those repressors get removed and we're free to make our protein. Now, the opposite form of this is going to be like the trip operon, which is going to be involved in tryptophan. So the default state for tryptophan is that it is on. It is constantly making, it is constantly transcribing the genes to make the proteins that make tryptophan. And so these are going to be frequently used, these repressible operons, in pathways that we use a lot. So if we're using this function a lot, we want this to be on all the time, and then we're going to repress it to turn it off if there's too much of something. So if we have too much tryptophan, what tryptophan will do is bind to our repressor protein, which is naturally in the off position. That causes a conformational change that is going to make it so the repressor can bind to that promoter region and block RNA polymerase. Now, why would you want to do this? Well, if you have too much tryptophan, there's no point in using cellular resources to make even more tryptophan. So once you have enough of it, shut it off until we need more tryptophan, and then we'll turn it back on again. So when that repressor is inactive, that means we are really low on tryptophan because there's no tryptophan binding to the repressor. So we're going to keep making all these proteins that build tryptophan. Then when tryptophan levels go up, because we've built a bunch of tryptophan, we have excess tryptophan, it's going to bind to our repressor, which is normally in the off state, and it is going to cause that conformational change to bind, and now that'll turn our operon off. RNA polymerase can't bind, shutting off production. And so just think about it as if you have a lot of something, why would you get more? There's also something called upregulation, and this is going to be referring to increasing the baseline amount of transcription through the binding of an upregulator. So this is going to be something that can bind to that promoter region and cause even more of that that protein to be produced. Now, let's get into our eukaryotes, because there's going to be more complex controls with eukaryotes. We can affect things with access to DNA. There'll be pre-transcription and post-transcription forms of control. There will be post-translation forms of control, and then chemical signaling that will play a role. So let's start off first with access to DNA. And we're going to need to talk about how our DNA and chromosomes are built. So DNA is wrapped around histone proteins. It's really tightly wound around these histone proteins, which group up into these clusters of 8 called nucleosomes. And this is called chromatin, which is super tightly condensed DNA. But when it's super tightly condensed, our RNA polymerase isn't going to have good access to it when it's wrapped around all these beads. And so that's going to effectively turn off a gene. So we need to loosen DNA in order to have access to it. So there's going to be 2 forms of groups, acetyl groups and methyl groups, that are involved in whether DNA is tight and turned off or loose and open for business. So if we add an acetyl group, which if you think back to biochemistry when we learned those functional groups, this is going to cause proteins and the histones to loosen up. It's going to cause them to unwind and expose these sections of DNA where genes are, so it can be read and be open for transcription. When it's super tight like this, it can't be read. Methyl groups, on the other hand, are going to cause these histones to wrap up tight again. So acetyl groups will loosen DNA, methyl groups will tighten DNA. Methyl groups tighten DNA and block access or transcription, and acetyl groups loosen or relax our histones in DNA, which allows the gene to be active and accessible by things like RNA polymerase. So here we see our methyl group blocking transcription and acetyl groups, which are facilitating transcription. And the really cool thing about these is that this is how we differentiate between different cells in our body. So your stomach cells don't need to make neurotransmitters that your brain makes. And so it has the genes to do that, but they're heavily methylated in the stomach so that they're never transcribed and we don't make any neurotransmitters. Those same genes will be acetylated in the brain so that we can make those neurotransmitters. Conversely, the stomach will have acetylated genes for making stomach acid, whereas in the brain, you don't want acid eating away at your brain cells. And so those will be highly methylated in the brain. Another cool thing about methyl groups is you can actually change your methyl groups over time. All sorts of things you do in your life, things like what you eat, what you drink, what you smoke, pollution that interacts with you, any sort of thing that we've talked about that may be like a carcinogen that could cause cancer, can actually methylate your DNA and turn off certain genes. And if this occurs in the sperm cells of a male during their lifetime, that can result in those methylated genes getting passed down to their offspring. And so what you do in your life can actually be inherited later on. And so sort of causing a whole turn on the head of what we've understood about evolution and heredity. So the world of epigenetics is really fascinating. So pre-transcription. Now, eukaryotes are going to control transcription through different things called transcription factors, which are a way that you can either upregulate or downregulate certain proteins. So there's all sorts of influencers. We've already talked about a promoter when we were talking about prokaryotes, and this is the sequence in front of a gene that's recognized by RNA polymerase and initiates that binding. We talked about that in our transcription and translation with things like the TATA box and things like that. And then there's things called enhancers. Now, enhancers are sequences that are even further upstream from the gene, so before the gene in the DNA, that are going to increase transcription by binding transcription factors and then folding them over to the promoter region. So what you can see here is that our enhancer is going to grab onto transcription factors that initiate transcription, and then it will fold itself over. That's why it's ahead downstream. It's far enough away that it can fold over, and you can think of it as grabbing on to that transcription factors and then just pushing that onto the promoter region to guarantee that transcription happens. So those enhancers are just going to boost the odds that those transcription factors bind to that promoter. So here we can kind of see that broken down where we have a DNA strand, and way upstream we have our enhancer. Downstream of that we have our promoter and our gene, and then we have all these transcription factors and RNA polymerase that are going to be necessary. And so this enhancer grabs onto those transcription factors. That's going to then bend over and bind to the promoter, which allows RNA polymerase to start transcribing that gene. All right, transcription has happened. Now what can we do to control our genes? Well, remember we talked about splicing in transcription as part of that final stage of transcription. And there's different parts of the RNA that's made. There's exons and introns, and remember the introns get cut out, and the exons are what stay and are included in that final protein. And with a lot of genes, there's something called alternative splicing. So different exons will be included in alternate forms of a gene. So not every gene will be exact, or not every protein it makes will be exactly the same based on if it needs slightly different functions. And this allows us to conserve DNA when most of a protein is the exact same. It allows us to use the same stretch of DNA for multiple different versions of a protein, which creates some really great opportunities for multiple forms of a protein. And then finally, there's something called RNA interference, which if RNA is being made, that RNA can be cut down or degraded so that it's not going to continually be produced. And this is important because if mRNA is made, we don't want it to just keep getting read over and over and over and over again. We want to make sure that we can break down that RNA and that it's not getting overly used all the time. And then that will prevent translation from happening. All right, translation has occurred. What can we do after the protein is made? Well, there's protein targeting. So this is signal sequences on polypeptides that target where a protein ends up. So this will be a little sequence that actually gets removed from the folded protein, but it's important in things like the Golgi body and the endoplasmic reticulum in sending that protein where it's supposed to go. Because sometimes very similar proteins will perform different functions if they're inside the cell, outside the cell, or sent to a different cell. So this is going to also affect in how our genes express themselves. And then finally, we can make proteins that aren't turned on. They will be turned off. So sort of like when we looked at that operon and we saw that there was a protein that was deactivated in the inducible operon, where it's deactivated until it's turned on to activate that, to turn off that operon. And so certain proteins are just waiting for some sort of cellular pathway or some sort of signal to turn them on. And so then they can start getting used. So just making a protein doesn't always mean we're going to see that protein in action right away. And a big part of how we're going to regulate that is through something called chemical signaling. So if you remember back to when we talked about membranes, there's receptors on the surface of our membranes. And those receptors bind to a signal called a ligand. Now, there's all sorts of different chemical ligands. The most commonly known one that you may know of is like hormones are a form of a ligand because they're a chemical signal that'll bind to a receptor. And so basically, our signal transduction pathway works like this. Without any ligand, our receptor is off. And so we have no response. When a chemical ligand binds to that receptor, that's going to turn that on. Now that receptor is going to turn on another protein at the membrane. That protein will then activate protein in the cytoplasm, which will activate a final target protein in the pathway, which will initiate a response. Now, what kind of responses could we be looking at here? Well, this can be involved in gene regulation. It's possible that signal transduction, so we saw that the ligand binds to a receptor, that's our reception. Transduction is passing from protein to protein. Sometimes it's multiple proteins. Sometimes lots of proteins amplify the signal. And then we get into a response. Sometimes that final protein that gets activated is turning on transcription of a gene. So it's initiating protein synthesis and changing how we express a gene. Sometimes, like those inactivated proteins I was talking about a minute ago, will get activated. So that final protein will bind to an inactive protein to turn it on. So this chemical signaling can be both affect pre-transcription and post-translational controls for gene expression. So here we see an example of growth factor, which we talked about that signal when we were talking about cell division and cancer. It binds to the receptor, goes through the transduction pathway, which turns on transcription factors that will activate the cell division promoting gene that will make proteins that then cause growth and division. So we can see that that's only happening. These proteins are only being made once those transcription factors are activated by the chemical signal from growth factor. So reception, transduction, response. And these that transduction system relies on something called second messengers. And these are internal signaling molecules. They could be chemicals, they could be enzymes and proteins. And what they do is they relay the message from that receptor to the nucleus or wherever our response is going to be. So cyclic AMP, as you can see here, is a form of that. There's all sorts of enzymes. We've talked about like kinases, those enzymes that attach phosphate groups to other proteins to activate them. That could be in this transduction response. And this transduction frequently has something called amplification. So when our ligand binds to our receptor, it's not receptor, one protein, then response. That reception protein may activate 5 different proteins or second messengers, and each one of those may activate a subsequent 5 second messengers, and each one of those may activate a second 5. And so you can see how that from one ligand, all of a sudden we have a whole bunch of second messengers or chemical messengers flooding through the cell to turn on whatever our response is. So turn on that transcription. And why would this be important? Remember, the cell can't think, and these chemicals don't know to go to certain places. And so by amplifying, when these chemicals spread out in the cell through diffusion, it means it's more likely that we'll turn on what we're supposed to be turning on. And it'll also happen a lot faster because there's more chemicals bouncing around. One of them's going to hit their goal sooner. And when we talk about these cell signaling pathways, this is a huge area of study for medicine, is how we turn on and off certain genes and proteins through these signal transduction pathways. So for example, birth control, we use hormones to make sure that ovulation does not occur. Antihistamines, when you have a cold, is responsible for blocking a histamine receptor, which signals a whole immune response so that you don't have that stuffiness and those overwhelming immune responses. By blocking those receptors, we don't have the transduction pathway and we don't have that cellular response. So really cool and practical stuff that we can see there. So big take-home ideas for you. Make sure you understand operons, both inducible and repressible. The AP test loves these things. Understanding that access to DNA with those epigenetic controls, so methyl tags causing the histones to close down, blocking transcription, and the acetyl groups that allow it to loosen up so that DNA can be read. Understanding those different levels of eukaryotic gene control. So understanding that there's pre-transcription, post-transcription, post-translation, access to DNA and chemical signaling. You don't need to know every single detail there, but you should be familiar with alternative splicing. You should be familiar with like those introns and exons. You should be familiar with that there are things like enhancers that will bind to the promoter, like tagging those proteins for where they're supposed to go, or also the activation of proteins. And then finally, cell signaling. We didn't really cover it that much in membranes, but I do want you to understand those layers of reception, transduction, and response, that hormones and other chemical signals can cause a transduction pathway to occur in a cell that can bind to a gene to turn on transcription, or activate one of those proteins that we had already ready to go, but in an inactive state. All right, hope you learned something, and we'll see you next time.